Jack, and this
Hello,
is
and my name is Fabian. Today we're going to share about our DNA president presentation slides, science fair.
okay. This is the
This is
purpose
the
of
purpose of the
this.
DNA.
The purpose of this research is allows us to understand DNA more better and its structure, like almost everything of it. And
Yeah.
this is a picture. Okay, this is a picture that allows us to know about the chemical structure of DNA and the in the chemicals inside it. This is
Pur- Purpose and background. So, the first purpose of our project is because DNA is the thing that has your characteristic in your blood. Second, DNA is one of the four major types of macromolecules that are known to be essential for all forms of life.
okay. So this is our first key
Concept.
concepts. Nucleobases, which is known as nitrogenous bases, or often simply bases, are nitrogen containing biological compounds that form nucleotides, which in turn are components of nucleotides, with all of these monomers constituting the basic building blocks of nucleic acid. It is one of the main parts that create DNA.
Key concept 2. Our key concept 2 is sugar phosphate backbone. Sugars that have added or substitute phosphate groups are often used in biological systems to store or transfer energy. They also form the backbone for DNA and RNA. Sugar phosphate backbone geometry is altered in the vicinity of the modified nucleotides. From the name, we know it's the bone for DNA.
Okay, this is the picture of the website that we use to research. So as you can see here, they are pretty a lot of chemicals and structures about DNA. And the recommendations. During the research, our team found a related topic that is called DNA nanotechnology. We all agree that it seemed pretty interesting. We could use that to find the we could use that to keep on researching about DNA.
So this is the work site. You can check out this link, and it was published in February 1, 2021 by Yasha. You can read it.
Yeah, shall we, Sharma? And we have one thing to show you is our DNA document
Our
about.
DNA research document.
Uh, this is our DNA research. Well, this is the basic information. DNA stands for deoxyribonucleic acid, which
Which
is a
is a
molecule.
molecule that contains the instruction an organism need to develop, live, and reproduce.
These instructions are found inside every cell and are passed down from parents to their children. That's why we say that they store characteristics in our blood.
It is also a nucleic acid and is one of the four major types of
This is
macromolecules that are
already this
that
has
are
already been in the
this
presentation. You could
yes.
still read it if you want to.
So of course, basically, the DNA is composed of deoxyribonucleotides.
And one thing, DNA is found in the nucleus with a small amount of DNA also present in mitochondria in the eukaryote. Okay, this is the picture from our presentation.
So a
And
short history of DNA. In nine in 1953, James Watson and Francis Crick discovered the structure of DNA. The works
it,
of
So
Rosalind Franklin lead to Watson and Crick's discovery. Franklin first had point out that the DNA is made up of two spirals.
the structure of DNA looks like a double helix structure because it looks like a twisted ladder. The sides of the ladder are made of alternating sugar, deoxyribose, and phosphate molecules, while the steps of the ladders are made of a pair of nitrogen bases. There are four types of nitrogen bases: adenine, which is shortly called A, thymine, shortly called T, guanine, shortly called G, and cytosine, shortly called C. This is the DNA pairing. The nitrogen bases have a specific
Specific.
pairing pattern.
This pairing pattern occurs because the amount of adenine equals the amount of thymine. The amount of oh my.
Guanine is equals to the amount of cytosine. The pairs are held together by hydrogen bonds.
Each strand has a 5 end with a phosphate group and 3 end with a hydroxyl group. The strands are antiparallel, meaning that one strand runs into 5 to 3 direction while the other strand runs in a 3 to 5 direction. Types
These
of
are
DNA.
These are types of the DNA strands. There are two DNA strands that called polynucleotides, as they are made of simpler monomer units called nucleotides. Each strand has a 5' with a phosphate group and a 3' with a hydroxyl group. The strands are antiparallel, which meaning that one strand runs to a 5 to 3 direction while the other strand runs to 3 to 5 direction. The two strands are completely held together by hydrogen bonds and are complementary to each other. Well, this is from up top here. You guys could skip this part. Major and
Minor
minor
groups of
groups of
DNA.
DNA. As a result of the double helical nature of DNA, the molecule has two asymmetric grooves. One groove is smaller than the other.
This asymmetry is a result of the geometrical configuration of the bonds between the phosphate, sugar, and base groups that force the base groups to attach at 120-degree angles instead of 180-degree. The larger groove is called the major groove, occurs when the backbones are far apart, while the smaller one is called the minor groove, occurs when they are close together. Since the major and minor grooves expose the edges of the bases, the grooves can be used to tell the base sequence of a specific DNA molecule.
The, poss- the possibility for such recognition is critical, since proteins must be able to recognize specific DNA sequences on which to bind in order for the proper functions of the body and cell to be carried out.
Properties of
Properties of
DNA.
DNA.
DNA
DNA helix could be right-handed or left-handed, but the bead conformation of DNA having the right-handed helices is the most stable. On heating the two strands of DNA separating from each other and on cooling these again hybridize.
The temperature at which the two strands separate completely is known as melting temperature, short as Tm. Melting temperature is specific for each specific sequence. The B sample of DNA having higher melting points must have more Cg content because Cg pair has 3 hydrogen bonds. The sequence of bases along the DNA molecule encodes for the sequence of amino acids in every protein in all organisms.
Types of DNA: eukaryotic Organisms such as animals, plants, and fungi store the majority of their DNA inside cell nucleus and some of their DNA in organelles such as mitochondria. Based on the location of DNA may be
Locate within the nucleus of nuclear DNA. Located within the nucleus of eukaryotic cells. Usually has two copies per cell. The structure of nuclear DNA chromosomes is linear with open ends and includes 46 chromosomes containing 3 billion nucleotides. Nuclear DNA is diploid, originally inheriting the DNA from two parents. The mutation rate for nuclear DNA is less than 0.3%.
and this is mitochondrial DNA. Mitochondrial DNA is located in the mitochondria, as the name, contains 100 to 1000 copies per cell. Mitochondrial DNA chromosomes usually have closed circular structures and contain, for example, 16,569 nucleotides in human. Mitochondrial DNA is haploid, come only from the mother. The mutation rate for mitochondrial DNA is generally higher than nuclear DNA. Forms of DNA. Most of the DNA is in the classic Watson-Crick model, simply called as a bDNA or B-form DNA. In certain conditions, different forms of DNAs are found to be appeared in, like aDNA, zDNA, cDNA, dDNA, eDNA. This deviation in forms are based on their structural diversity. Number 1, bDNA. Most common, originally deduced from X-ray diffraction of sodium salt of DNA fibers at 92% relative humidity. 2, which is aDNA, originally identified by X-ray diffraction of analysis of DNA fibers at 75 relative humidity.
3. zDNA. Left-handed double helical structure winds to the left in a zigzag pattern. 4. cDNA. Formed at 66% relative humidity in a presence of L plus and Mg2 plus ions. 5. dDNA. Rare variant with 8 base pairs per helical turn. Formed in structure devoid of guanine. 6E DNA. Extent or eccentric DNA.
Functions of DNA. DNA has a crucial role as genetic material in most of living organisms. It carries genetic information from cell to cell, from generation to generation. Thus, its major functions include storing genetic information, directing protein synthesis, determining genetic coding, directly responsible for metabolic activities, evolution, heredity, and differentiation. It is a stable molecule and holds for more complex information for longer periods of time. We thank you for listening to our presentation, and goodbye.
Goodbye. Thanks for listening.
Thanks for listening.